Now, the last thing I want to talk about is the idea of a reducing sugar. As we go on, we're going to look at more complex sugars, and we're going to be looking at them in their ring format. So again, I'm going to remind you, this is the basic backbone of a six-membered ring. And remember, we always have our CH2OH group here, and we have some OH groups that are pointed in different directions. I'm just happening to draw glucose. And our new OH group that forms to the right of the oxygen where we have cyclized. This sometimes can be represented as a squiggly line to represent that it could be in the up position or the down position, or again, it could be the alpha or the beta form. Because this is variable, because this can happen in either way, sometimes we just draw this OH again with a squiggly line just to say it could be either. You might be given a drawing where it is up or it is down and asked to assign its name as alpha or beta. If it doesn't have an up or down positioning, you don't assign an alpha or beta to its name. Now, this OH group, again, as I said, is very special, and it is on a carbon that is called the anomeric carbon. That's the special name for this carbon where our new chiral OH group forms in the alpha or beta position, the anomeric carbon. Now, when we start linking together sugars, which we'll talk about in the next section, you'll see that this anomeric carbon, this OH group, is responsible for participating in the linking of sugars together. If this anomeric carbon, if this OH on the anomeric carbon is free, meaning it does not have any bonds to it, if it looks like I have drawn it right now, where it is free, we call this a reducing sugar. So the part of a sugar or any section of a sugar that has a free anomeric carbon is classified as a reducing sugar. This is important because some sugars, when they start to bind and make complex forms, become non-reducing. They do not have this free spot for binding. And we can detect if sugars do or do not have an open anomeric carbon, or if they are a reducing sugar. Some of the detection methods utilize metals. One is called the Tollens Test, which utilizes copper. And as you can see, it's going from a change in charge. That's where this name of reducing sugar comes from. It can reduce, it can undergo this oxidation reduction reaction with a metal. Similarly, you can use the Tollens Test, which does the same thing except utilizes silver here, going from silver +1 to neutral silver. Again, an oxidation reaction, and the change in this charge represents that you do have a sugar with a free anomeric carbon. You have a reducing sugar. There are other types of techniques to determine if this reducing sugar is present, and those are colorimetric, meaning they're a technique that utilizes a change in color for us to tell if it is free or not, or they are an electrochemical test. Again, they're going to be utilizing some change in charge, but that charge will be then used to conduct some electricity to say yes or no. For glucose and other reducing sugars, there's also enzymatic methods that can be used. this is a type of method that is colorimetric, one that changes colors. Here we can see that glucose, when combined with oxygen and this special enzyme, remember enzymes catalyze reactions, will make a new structure that is a lactone. We just looked at an example of this. This is glucanolactone. But the key part here is that when this reaction happens, it produces H2O. So for this lactone to form on an anomeric carbon, it has to have a free anomeric carbon. It has to be a reducing sugar for this reaction to even occur. So unless there is a reducing sugar present, H2O2 or hydrogen peroxide won't form. If hydrogen peroxide forms, then another chemical that's also mixed in this will be reacted with another enzyme, peroxidase. So this is a dye that is a faint orange. But if hydrogen peroxide is around, the dye will react and form this oxidized form, which is bright orange instead. So essentially, this all happens very fast, right? This chemical reaction occurs very quickly. But essentially, if you put glucose in the presence of this dye and you put glucose oxidase and peroxidase in there, so they're in the mixture, and you put your dye in, if glucose goes into that dye, this reaction will happen quickly. Hydrogen peroxide will be formed. Hydrogen peroxide will react with the dye, and it will turn it into a bright orange color. And you can sense, based on the color, how much glucose is there. Now, in something like one of those detectors where people are measuring their glucose, for example, if you're diabetic and you use a small detector to see your glucose content, this is going to be using an electrochemical detector for the sensor instead of a dye. So it's actually going to be looking for a change in charge that then conducts an electrical signal. And that quick electrical signal will be detected on the sensor, and that's how you can tell your glucose level for diabetics. So this type of dye reaction is usually used in a lab, for example, where we're not trying to quickly get some information in a small sensor and we have more equipment. You could use a dye instead. Okay, so that is it for the general outline of the structure of our carbohydrates, naming carbohydrates, and the cyclization of carbohydrates. So the key thing here is understanding how to go from a linear form to a cyclical form of a carbohydrate.